Then we talked about how oxygen is considered H2O2 by PBS1. 50% of oxygen will form superoxide molecule, which is phosphorus. And so this all has certain mechanisms to protect from that. So mitochondria and new SOD, superoxide, which decays and it converts that to H2O2. And the cytosol has the CUN SOD, which does the same thing but in the cytosol. And then, sorry, we have glutathione, so thionine is thione, sorry, which is utilized to neutralize H2O2 into water. So that is then oxidized in terms of the GSSG, which can be utilized in water. And then it converts back into DHS by a glucose reactase utilizing an ADPH and protein response. And then we talked about the late response.
What is that?
So when you talked about the late response, you said that it was the fusion of egg and sperm would lead to the formation of DAC, and then the activation of PKC with a large level of calcium opens the NAH or ion antiporter, which increases the alkalinity of the cell, and then exposes the nuclear membrane to break down and for DNA synthesis. And then we talked about calcium being a critical role for new DNA synthesis, and inactive MAP kinase leads to DNA synthesis. And we talked about protein synthesis starting, and then it can only start from existing mRNA in the egg, and there's no new mRNA synthesized. And we talked about histones, tubulin, and morphogenic factors. We talked about how on mRNA there is the translation initiating factor E, IF4E, needs to bind to the 5' end, but there's an EIF4E binding protein that blocks that from happening. So the increase in calcium activates CAMP, which activates protein kinase A, which phosphorylates the binding protein. And when it's phosphorylated, it's able to leave, and then it's degraded. And so then EIF4E, translation initiation factor, can bind and start transcription.
Oh, then what happens?
From the mRNA, cyclin B is also made, which interacts with CDK1, and they make mitosis promoting factors that start the cell cycle. And then during the fusion of genetic material, once the egg and sperm fuse, the flagella and mitochondria from the sperm degenerate. And then the sperm nuclei undergoes vesiculation because the CAMP activates the PKA, which phosphorylates the nuclear lamin, which causes it to disintegrate and leads to the vesiculation. The PKA also phosphorylates the protoamines, and basically repels them off the phosphate group, and then those are replaced by histones, which leads to the decondensation of sperm chromatid. And then when they fuse, once the nuclear membranes are gone, the centrioles duplicate and the nuclei shifts 180 degrees so that they're on either side of the cell, and then create spindle formations to create new cells.
Okay, so that was about the fertilization in the CLG. Now we are on the next topic, and we are going to talk about internal fertilization. In mammals. Okay, this is our next topic. And the fertilization, the study of fertilization, and even the development, everything is very difficult in the mammal. So it's very difficult to study. I purposely made it a subtopic because it is actually very difficult to study. So think about that mammalian fertilization occurs in oviduct. So that is inside the oviduct, and oviduct is inside the female. Okay. Now, we can, if we are studying the fertilization in sea urchin, you can easily mimic the condition for sea urchin fertilization, right? You can have the seawater, and you can make artificial seawater and put sperm and egg together, and you say, hey, we are mimicking that thing. But problem here is, for the mammalian fertilization, we still do not know the exact, what exactly there is. So, what is the environment? What is the exact natural environment? Of oviduct? We still do not know what is there. So if we do not know, that means, and think about oviduct is a long tube. So all the natural environment through which the sperm interact in the oviduct, we do not know what are those environments in the sperm is going to interact. Now, it not only interact that, then it has to reach to the egg also, right? It should travel that path, so we don't know what is happening here. And the other thing is, the most important thing is, when the sperms are released in the female reproductive tract, almost 280 into 10 to 6 sperms are released at one time, right? But out of these sperm, only 200 reach the vicinity of egg. Means 200 reach, only one can fertilize. Now think about, this is almost 1 in 10,000 sperms reaching there. So if you think about that, you had this many number of sperm, only 200 reaches near the egg. So it means there might be some molecules which are affecting only this many sperm to reach there. So it is very difficult to study those molecules which are regulating, like this many population of sperm is released, only this many are releasing. What is happening? What are those molecules which are allowing only this number of sperm to reach there? And then the other thing is that all of those sperm which are released at this point, so all these sperm which are released, they are not in the same stage of maturation. So some spermatozoa are mature than others. That means they are in different stages of development. So many sperms are released. Not all the sperms are in the same stage of maturation, and then out of these, only few released. So we don't know what are the molecules by which these sperms are interacting. And the other thing is, there can be multiple mechanisms of acrosomal reaction in mammalian sperm. So all those things that really make it very difficult to investigate the mammalian fertilization. So we are still trying to investigate, still people are trying to look at how that happens, okay? Now, having said that, but still we know a little bit, and what we know we want to talk about that. So then how the translocation of gametes takes place. Now, the female reproductive tract is very important. Now, this female reproductive tract is not a passive conduit. It's just not a passive conduit through which the sperm will enter and run away. It's not like that. So, female reproductive tract is highly specialized tissue that not only regulate the motility of gametes, but also regulate the maturation of gametes. And those gametes are both oocyte and sperm. Both. It's not only sperm or only oocyte. So, then what is there? Both male and female gamete, what they utilize small biochemical cues, and large physical propulsions. To translocate and mature. So physical propulsion takes place as well as small chemical reaction takes place. Now what is happening there? So if I'm going to draw the structure, so this is the structure of the female reproductive tract. So right here is uterus. Here is vagina. And from here, this part, whole part is essentially oviduct. Okay, this whole is oviduct from here to here. And this part, these are the fimbriae. Right, this part is called isthmus. This part right here, this one part is called ampulla. This is ampulla, and right here is infundibulum. Okay. So these are the different parts. Okay. Now, what is happening there? So I use the term that this part is not a passive conduit. It regulates how the sperm and the oocyte is going to enter here. Okay? And right here is the place, this is the ampulla. This is the place actually where fertilization takes place. Okay. Now, you can think about that the sperm enters here, it needs to reach here, right? And then we are saying that right here is the ovary. Remember that from ovary, you have that one oocyte ruptures out, right? And when that oocyte ruptures out, so this is your oocyte, and that oocyte we know is surrounded by cumulus cells. So these are the cumulus cells. Now, this ovary, when it releases a oocyte, when the Graafian follicle matures, right, and this is released out. This is captured by the fimbriaries of the oviduct. Now, the most important thing here is that this fimbriary is going to pick up this oocyte only if the cumulus cells are there. If you remove the cumulus cell, then this fimbriary cannot pick up the oocyte. So that's the critical thing. So you have this thing, this is released here, this oviduct is right there, it essentially catches up with the fimbriary, and now this oocyte is right here inside. With all those cumulus cells. I'm just drawing one there, but there are many layers, you know that, right? Now, the physical propulsion, the movement of the muscle, that causes this to reach here. Okay? Now, what is going to happen? So, the oocyte has reached its destination where it should be, where the fertilization is going to take place. Now the sperms, which are released here, so sperms moves to oviduct by physical propulsion. And that physical propulsion is the muscle contraction of uterine muscles. So uterine muscles, they contract here and they allow the sperm to reach here in this area of the oviduct. Okay? Now, they have to reach the ampulla. But what happens, so first this propulsion move it here. Right here, the sperms are slowed down. So they are not allowed to run here. So sperm in the oviduct are slowed down. And slow down before reaching ampulla. So they are slowed down right here. Okay? So now what happened? The sperms are slowed down here. From here to here, when the sperm are being to move, at that time, so here was the muscle contraction, but from here to here, they require flagellar movement. So flagellar movement is required to move in the oviduct. Okay, so they need some sort of flagellar movement. Now, then where to go? How does this sperm know it need to come here instead of going that direction, right? So that the sperm, so there should be directional cues. So sperm receives directional cues from the gradient of temperature in the different parts of oviduct. So different parts of oviduct have different temperature. That is first thing is it is sensing. And then there are chemical cues. There are chemical cues released from the cumulus. There are chemical cues released from the cumulus. So they are telling them. Now, the other thing what happened during this trek, so during the trek from vagina to ampulla, sperm gain the capability to undergo acrosome reaction and fertilize the oocyte. So that is when the sperm is released, it does not have any capability to undergo either acrosome reaction or undergo fertilization. So that means what happened, all these capabilities are making the sperm mature. Sperm is getting mature. And it gained the capacity or the capability to undergo acrosome reaction and fertilize the egg. Now, this capability, so sperm when it was released, it didn't have that capacity or capability to fertilize the egg. That maturation process, which is achieved by the sperm during its trek in the oviduct, is called capacitation. Is called capacitation. So capacitation is that the set of physiological changes during the sperm trek in the oviduct that provide the capacity to fertilize egg. So here is the important thing. So what we learned so far. So we are saying that the female reproductive tract is not a passive conduit. It's a very active tissue. And that very active tissue releases various biochemical cues and the physical cues. Those are taken up by both the gametes, and those cues help both male and female gametes to translocate and mature. And we are saying that when the ovary release the oocyte, those oocytes are captured by the fimbriae of the oviduct. If you remove the cumulus cell from there, then these fimbriae, they cannot catch the oocyte. That means that these cumulus cells are critical for that process. Then the physical propulsion set it from this area, the infundibulum, to the ampulla. Okay? Now, sperm which are released here, they are diverse type of spermatozoa. They are in different stages of development. Out of so many sperm which are released, 1 in 10,000 can only reach near the egg. Okay? And then when we are saying that what is happening, that means sperm are getting physical as well as the biochemical cues during this process, and this female reproductive tract is actually providing some cues which are allowing this sperm to reach here. And those cues are temperature gradient. There are temperature gradient that sperm can follow. And then the other thing is the chemical released by the oocyte and the cumulus that can also provide a direction to the sperm where to go. Now, then we are saying that the sperm during this track, they gain the capability so that they can undergo acrosome reaction and fertilize the oocyte. So we are saying those physical and biochemical changes which take place in the sperm during this track in the female reproductive tract, those make it capable to fertilize the egg, and that process is called capistation. That is clear? Okay, can talk to each other for a minute, and we'll go further.
It's like the end part. It's like this part. It's just like this section. So not know the exact nature or natural environment of the duct which is from interaction with the duct. But the sperm releases about 280 pounds in the six. And at one time when it's able to reach the vicinity of the egg, that's about 1,000. And then out of the 200 in the transmitters, they're not all in the stage of maturation. Some are more mature. There could be multiple mechanisms of the acrosomal reaction in the mammalian sperm. Through translocation of the gamete, like I said, the female reproductive tract, highly specialized tissue, not only regulate motility of the fetus, but also regulate the maturation of the gamete, both the oocyte and the sperm. And utilize small biochemical cues and large physical propulsion to translate and mature.
Then the ovary, we have, or we have the ovary that has the oocyte, and then that process with the cumulus layer of interface, which is caught by the fimbriae, and then the muscle contractions causes it to reach the fertilization center and all that. And the sperm is in the uterus and used in the oviduct by propulsion, or
Physical propulsion.
by muscle contraction, underlying muscles. And then in the oviduct, they are slowed down before reaching the ampulla, and they use flagellar movement. Then sperm receives directional cues from the gradient temperature and different parts of the oviduct, as well as chemical cues released from the cumulus, and then urinary tract from vagina to the ampulla. Sperm gain ability to undergo acrosome reaction and fertilize oocyte. This means the sperm matures, which is called propulsion. Because of physiological changes during sperm tract and
Any doubt here?
its capacity to fertilize the egg.
Any doubt? Okay, now. So what is happening that the sperm, those are released from here, right? Those sperm are actually stopped right here at the isthmus. So all the number of sperm which are released here, they are actually stopped at isthmus, and all the sperm, so you can draw this thing again. So all the sperm, they are attached here to the isthmus. So the sperm which are released here, remember in the uterus you have that muscular propulsion that throw them in the oviduct, and the sperm, they are actually attached here to the sperm. So all these sperm are not ready, yet ready to fertilize. So what happened? That uncapacitated sperms are attached to isthmus. Okay. So we are essentially talking about sperm translocation and capitation. This is what we are actually now about. Okay. So now, if we are saying that what is capitation, there are two things which should happen in the capitation. So capitation, it prepare for two things. Number one, acrosome reaction prepares, and number two is these sperm should gain the motility. They should become hyperactive, right? And hyperactive means their flagella should start moving like crazy so that they can go to the egg. So second thing is making them hyperactive. Okay. So the fertilizing sperm, the sperm which have the capillary, which is actually going to fertilize, can take as long as 6 days to reach the egg. So it means that journey which that sperm is going to start from this part to the ampulla is going to take 6 days. So those 6 days are going to prepare it for fertilization. Now, if we are saying that the capitation is preparing it for acrosomal reaction, that means these events are taking place at two places, capitation events. And they are taking at different times. Number one is the events at sperm head. The sperm head events are going to prepare it for the acrosome reaction. And the events which are taking place at the tail, at the sperm tail. And those events are telling it to provide hyperactivity. Now, both these events are occurring at different times. Occurring at different times. Okay, now, if you look here, this is the cow isthmus. So you can see here this sperm is kind of attached to the isthmus. This is in the cow. Okay. So it's so down here. So then how this bonding is bound now? How you're going to break this bonding, right? We said uncapacitated sperm are attached here. When the sperm gets the capability, when sperm is capacitated, what is going to happen? That it should show hyperactivity. Once it becomes hyperactive, then it is released from the isthmus. So uncapacitated sperm cannot be released. Once the sperm gets capacitated, then the sperm is released. And which part of the capitation? Hyperactive. So hyperactive sperm is released from the isthmus. Okay, now, what is the purpose of this point? It serves three important purposes. Number one, so we are saying purpose of sperm binding to isthmus. Three purposes are there. Number one, prolongation of sperm's lifespan. Okay. Now, these sperm which are slow down, which are attached here, so we said millions of sperms are released. You don't want all those sperm to capacitate it at the same time. If they capacitate it, they are done, right? So it means by attaching them and by delaying their capacitation, we are prolonging their lifespan. Making sense? Now, there are some speedy sperm which can reach from here to here in 30 minutes. We call them speedy sperm, but they can never fertilize that. So it doesn't mean if sperm reaches from here to here, it can fertilize that. No, they cannot. They reaches within 30 minutes, but they cannot fertilize that. So we said it should be at least 6 days to get that capability. Now, so first thing is we are prolonging the lifespan. The second thing is what we are doing is we are preventing polyspermia. Because we are blocking the huge number of sperm to reach here at one single time. See, think about if all these sperm are capacitated at the same time, you can have more and more sperm there. And they have actually done experiments in cows. They have actually removed the isthmus and they found the polyspermy cases there. So you prevent the polyspermy there. That's the other aspect there is. And the third thing is, what you are doing is, you are releasing the sperm, releasing sperms in small groups, in smaller groups. If you are doing that, what you are doing? You are having a bigger time frame, right? So say 10 sperms are released today, 10 sperm are released after today. So what you are doing? You are having longer available time, therefore more chances of fertilization. So you can have the sperm attached there and they can be released slowly in the packets, and then you are giving a longer window for sperm to meet egg or oocyte in the embryo. Making sense? So these are the three things. So what we said that sperm, they are not capable of fertilizing the egg, and that capacity which they gain is gained because we slow down them here. And that capability is essentially to undergo acrosome reaction and become hyperactive. That means the mechanism or the events which are taking place to prepare the sperm are happening at two different places, in the sperm head and in the sperm tail. Now both these events are occurring at different time points because you have to do acrosome reaction when you are near the egg. You have to do hyperactivity from here to reach the egg, right? So different times. And then we are saying isthmus, stop it here. The purpose here is to prolong the lifespan of the sperm, to delay the capacitation of the sperm. So all those sperms are not capacitated at the same time. And prevent the polyspermy. And then also, when you release those sperm in the small packets, you are giving the larger window for this sperm to reach here and fertilize the eggs in the embryo. That is making sense? Okay, so talk to each other for a minute, then we'll go further.
These are all uncontaminated sperm attached to this one. It should be all of them. And then cavitation mechanisms, which have hyperactive sperm, tail. Fertilizing sperm can take as long as 6 days to reach the ooze site. And then this is because of events at the sperm head and sperm tail occur over time. And then when the sperm is captated, it is hyperactive, it is released from isthmus. And the purpose of binding to isthmus is prolong sperm lifespan, preventing polysperming because it blocks huge groups of sperm from reaching the oocyte at one time, and releasing sperm in smaller groups, which makes longer available time and more chance of fertilization.
Everything is okay here? Okay, now, how these things are happening? Okay, now we are trying to find out the mechanism, okay? So let's say, mechanism. Fluctuation. So we'll talk about events which are going to happen at tail and egg. But first we will talk about what happens both at tail and the egg at the same time. Okay. So what happened that the sperm membrane is altered in the oviduct. Sperm membrane is altered. How that is altered? There are albumin proteins in the reproductive tract. In the female reproductive tract, there are some albumin proteins. What they do, they remove cholesterol from sperm membrane. Okay. And when that happens, so now you have the cholesterol efflux from sperm membrane. I'm just writing as a. So what happened, what that does, you know about the lipid rafts or not? Lipid rafts, we know, we should have done in cell biology. What are those?
And they they're groups that move together and they have the proteins in them.
Yeah, they're
Okay.
like stiffer wraps.
Little more than that? Okay, so let me just explain it here a little bit so that you know. You should know.
Yeah, I remember vaguely, it's like groups of cholesterol that come around.
Let me, you are also going, but anyhow. So right here in the cell, in the membrane, usually you have phosphatidylcholine lipid very high, right? But then there is another lipid, what we call sphingolipid. Sphingolipids. Okay? these sphingolipids are the lipid. They have actually a very long tail of hydrocarbon. So this is a very long tail of hydrocarbon. So for example, this. Okay. And they also have a lot of cholesterol there. So this is all cholesterol. Okay. So think about some of the lipid rafts are here. Now cholesterol provides rigidity to the membrane, we know that, right? Lipids are kind of more of a fluid-like thing. So if you're looking this thing from here, have you seen the raft in the river? Rafts are usually like water and above something. That's why they call them lipid raft. Because of their longer tail, if this is the river, say they are kind of like above that. And these are the area where you can have this type of structure have the capability to carry many type of proteins with them. Okay? So they carry a lot of proteins there. And a lot of those proteins which are there, they are usually the different kind of transmembrane receptor proteins. Most of the time they are transmembrane receptor proteins. So, if you have, for example, this is your sperm head. I'm just drawing the sperm head, the whole sperm. Okay? So in this sperm head, for example, I'm just drawing here. So one lipid raft is here, one lipid raft is here, one lipid raft is here somewhere, one lipid raft is somewhere here. Okay? And right here, you have some cholesterol here, some cholesterol here, some here, some cholesterol here, some cholesterol here, some cholesterol here. Okay? So what you are doing? And then you have these lipid rafts, they are attached to certain receptor proteins. So you have these receptor proteins. You have this brown is cholesterol. And these are your, the blue are your sphingolipids. Okay? Now, so you remove the cholesterol. So that's what we are saying. We are removing cholesterol. Okay? So now you are removing cholesterol. What you are doing, when you remove the cholesterol, so these lipids are now free to move. Okay? So removal of cholesterol, what it causes clustering of lipid rafts. At the head of sperm. So then what is going to happen? That is going to lead to this thing. All those lipid rafts which were there, they are all going to come here. And because the cholesterol is gone from there, so these all lipid rafts, they are going to concentrate on one side, and they will have all those receptor proteins clustered together. Okay? So that causes clustering of lipid rafts. Therefore, clustering of protein receptors at the sperm head. Now, what is happening? That means if these receptors have those, remember we said that to bind the egg membrane, you should have binding, right? Binding should bind to something in the egg. So that means you are having all those receptors coming together. So you're providing, therefore, the proteins those are going to bind, those are going to bind to zona pellucida are clustered. Together. That's making sense? Now, other thing happened. This is okay so far? Now, other thing happened is that on this sperm surface, for example, right here, just think about right here. There was a protein, another protein. So there was another protein right here. There was another protein right here. Those proteins were maybe masked by, so think about this, these proteins, these green proteins are masked by cholesterol. Right? So when that happens, if the cholesterol is removed from here, these proteins are now unmasked. Making sense? So then these unmasked proteins are now also there. So these are the proteins there. Now this protein also might play a role in the sperm activation and sperm capacitation. Right? Now, so proteins are masked by cholesterol, and then proteins are unmasked. And if they're unmasked, they are helping to bind to zona pellucida. Now, other than that, cholesterol, this albumin protein, they not only remove cholesterol, albumin protein, they can also extract carbohydrates and other proteins. So think about, there is another class of protein here. Think about, there is another class of protein there. This is protein right here. Now this protein was kind of covered by, say this is a carbohydrate, and there was another protein attached to it, right? So if the cholesterol is removing this and this from there, so this protein is also exposed. So it means that protein is also unmasked. So you have all the protein are unmasked and go here.
Was that masked by a lipid or?
Carbohydrates.
Oh, carbohydrate.
Carbohydrates and the other proteins. So there can be some carbohydrates which can unmask it. So this is a carbohydrate. Just think about a carbohydrate.
And then albumin.
Albumin is going to remove that also. So that carbohydrate is now gone. Now this protein is unmasked. Protein. So all these activities are going to expose the proteins. Those can directly or indirectly bind to zona pellucida. Right? So if you give this chance to bind to zona pellucida, that means you're providing a chance for sperm and zona pellucida binding. Making sense? If that is going to happen, then you can have a acrosome reaction take place. So that's how, excuse me. So that is what is happening. Okay, so you guys are okay with this? So talk about this for a second and we'll go further. Any confusing? Any confusing? Okay, so. We'll stop here, we cannot do it in 5 minutes. So we'll keep on talking about this for the next, and we'll go from there. Okay? And then we will, yeah, we are meeting on Tuesday, that's